So, we will start again the major histocompatibility complex. Few important thing we need to know, and this will continue from last lecture also. So, we are going to discuss like what is the recognition of MHC TCR. We are talking about different domain like polymorphism, how it is related to interact with the TCR binding of the MHC part as well as the peptide part inside the MHC, how it is interacting. So, this interaction, I mean if you consider, if you understand like MHC with peptide and TCR how they interact. So, we have a better picture now. Okay. So, what we can tell is that this antigen presenting when everything is perfect, like a TCR is interacting with the MHC along with the peptide. You see in this cartoon, which is very straightforward, not like before the crystal structure, the MHC 1 alpha 1 and alpha 2 presenting antigen X. Here MHC A, whatever you are seeing, this is just to differentiate between two different MHC. You see MHC A here, MHC B here. It is not no relation with 1 and 2, all are MHC 1. So, this MHC 1 is present, MHC A type MHC 1 is presenting X antigen, and any particular T cell having a receptor like this is interacting both with MHC nicely as well as with the antigen. So, this is the ideal condition for TCR and MHC antigen complex recognition. But if the MHC is different, even the antigen is same, the X, okay, but TCR it can recognize X, but as it cannot recognize the MHC 1 or B type, there will be no interaction or no recognition. Same way, if this same MHC A type MHC 1 presenting Y molecule, because one type of MHC can present X, Y, Z different type, they are not that restricted, that we already know. In that case, this TCR which can recognize antigen X will also not recognize. So, that means the TCR recognition to antigen MHC complex is not either MHC or antigen independently, they should be both antigen and MHC complex, both should be recognized by particular T cell receptor. So, this phenomena is also known as MHC restriction. Okay. So, neither this case nor this case nor this case, neither one will have any recognition, and if there is no recognition, the T cell will not do anything, because this interaction, strong interaction is very much important. If this interaction happen, then only signal will go inside and give the cell response to do next job like proliferation, producing different cytokines to activate B cells and to get to have, as this is MHC 1, to be activated by the effector cells, so that it can kill the cytotoxic, these cytotoxic T cells can kill the tumor cells or virus infected cells, but if this one is not. So, these phenomena again I am repeating is called MHC restriction. Okay. So, we mostly whole immune system, in most of the immunology book, if you find, we are talking mostly about alpha and beta type of T cell receptor. Okay. Gamma and delta T cell receptor is not known as much as we know about the alpha and beta type. We know that it is there, we know their function, not much research has been done and not much is included in the book. So, in basic immunology concept development or the course, also we have, I mean we do not include the gamma delta receptor part that much, but few things we should remember or know, that gamma delta chains or the gamma delta T cell receptor is not recognizing that normal MHC 1, MHC 2 presented cells. They have a very specific different sets of receptor made antigen that they can. I will just give you one example here. There is a specific receptor called, this is specific ligand which is T22. I can give you a series or table which is available in net or in book, like okay, gamma delta receptor can recognize different type of alternative kind of receptor by which it is recognizing, and this X1, 1, 2, 3, 4, 5. So, there is no point of remembering all these names at this stage. If you are working or doing research on gamma delta receptor of T cells in future, if you are interested, then you will have to know that, like what is known, what is not known, and what you should do. But at this moment only alpha beta receptor, their concept, the immune system is developed, and you just for the information you should remember that gamma delta T cell receptor bears an alternative receptor that made up of the gamma delta chain that can recognize different kind of antigens, not the general antigen which is processed and presented by MHC1 and MHC2. So far just we told the MHC restriction. That means T cells should recognize the receptor MHC first and then the antigen. But what I did not tell that time, definitely I told you before that this MHC should be self MHC. Okay. Self MHC means my T cell receptor will recognize my MHC. Okay. That is how they can figure out which is self, which is non self. That is a kind of dress code, like military code. So, whoever having that cap is Indian, whoever having that cap is from other country. So, seeing the cap or seeing the jersey, so MHC molecule is just like a jersey that we discussed also before, to know from far, okay, he is my team member or member of other team, okay, or the player of other team. So, MHC recognition is very, very important. Okay. So, whatever antigen presented by my MHC, I am talking about my MHC, then only T cell receptor can recognize that. So, my T cell receptor will not recognize the antigen presented by somebody else's MHC until unless it is very same or similar. Okay. So, this kind of training is very important when T cell developed. But even after that, it was found that there is some alloreactive T cells which recognize non-self MHC. How it is determined? It is determined because if this is true that my T cells cannot, particularly the cytotoxic T cells are more important here. If cytotoxic T cells are not recognizing anybody else's MHC, then transplantation should not be a problem. Why we are typing MHC so much? Because if any organ from somebody else is transplanted to my body, T cell will not recognize them because MHC is different, and if MHC is different, then no immune reaction, but it does not happen. If tissue is not matched properly, if MHC is not matching properly or tissue typing is not good for whatever reason, the transplanted organ will be rejected by our body very short period. It is just like a regular immune reaction. That we will study a little bit more detail while we are discussing the transplantation in the latter stage of this course. But at this moment, if understand or believe me that if any organ, even a small part of skin from somebody else is transplanted on my body, it will be rejected within 7 to 10 days. Okay. So, all cytotoxic T cell will be activated and eat it. So, if that statement like my T cell receptor can recognize only my MHC, if it is 100% true, then it should not happen. If just to search that effect, like why it is happening, it was found that along with that self recognizing T cell receptor, self MHC recognizing T cell receptor, which we have or which you see, along with that there are another group of T cell receptor which is alloreactive. Alloreactive means which can recognize non-self MHC molecule. That type of alloreactive T cell receptor are basically doing the rejection of the transplant organ or tissue. It varies from 1 to 10% depending on the individuals, okay, but they are not less abundant. 10% is good enough. If you see the total number of T cell, present in our body, 10% is a good enough number, or even 5% is a good enough number. So, these alloreactive T cells. So, while we are studying the MHC and the T cell recognition or interaction part, at this moment we should remember, along with self recognizing MHC, we also have alloreactive T cell. Allo means can recognize some other allele of T cell MHC, which is not my cell. Alloreacting T cell recognizing non self MHC molecules. Generally the percentage varies, it is between 1 to 10% depending on the individuals. Okay. This is one more information you should remember. Another important thing, that T cell receptor responds super antigen. What is this super antigen? By name you can guess little bit. Antigen means which can induce the immune system, or which can immune system can recognize them and find them as foreign, and there are antigen which is even more powerful called super antigen. Okay, what is the super antigen? I will show you the picture, it will be clear very much. Okay. So, if you see this picture, normally what happen, this MHC2 you can recognize by alpha, beta and 2 chain, alpha and beta domain presenting one antigen. So, this is normal picture. So, if T cell receptor can recognize it. So, in that case, if you remember the MHC restriction, what is going to happen? This T cell receptor will recognize this part and also this part and this part through antigen. So, this can happen, and this is very specific. If you just go back or remember that MHC restriction slides, where we show that MHCA where everything is perfect. So, interaction is there or recognition is there. In one case MHC is not matching, another case antigen is not matching. Okay. In both case it has no recognition, but here what will happen, there are certain antigen which we call super antigen, they do not need the presentation by MHC, they are even smart enough. Okay, that is why they are super antigen. In case of bacterial super antigen, you can understand from this slide, like there are two types, one is bacterial, another is viral. So, not all antigens from bacteria super antigens, certain bacteria produce some protein which act as super antigen. Similarly, some viral proteins are act like super antigen. Why they are called super antigen? Because they do not need to be processed and presented by MHC. What they are doing? They have some sequence which nicely interact with the alpha 1 domain with super antigen, as well as beta chain, which is a variable beta domain, and this. So, this part without going to proper channel, they can interact sidewise, and this sidewise interaction, what they are doing, even antigen is not there. Suppose there is no peptide here, this red is not there. Okay. If that red is not there, if suppose there is no red here, if there is no red here, even after that it will interact with the MHC and T cell. Okay. They will bring them together and make a strong interaction. Cell like T cell or any other cell that we have in our cell, they do not have any eye, right? They see only by each other, they talk between each other, they give signal to nearest cell or the neighboring cells by protein-protein interaction only. So, as soon as this interaction happen, T cell will realize or think that some antigen is presented and I am interacting. Okay. Even there is no antigen, this interaction may be there. So, this interaction in presence of antigen or in absence of antigen will bring MHC class 2 and T cell receptor together, and T cell will think that enough binding with enough strength is there. So, signal should go inside and they will be proliferated, and that proliferation will increase the number. So, what you can understand? You can I mean you can guess. Okay, I will tell the answer after discussing the viral one. You see, the viral one is doing the same thing, but in case of viral one more thing is there. The viral one, the viral antigen should be integrated. This is one difference normally, and here you see interaction between alpha chain of MHC and beta chain of T cell, alpha chain of MHC and beta chain of T cell. In this case, what happened? Both are beta. So, beta chain of T cell and beta chain of antigen presenting cells. So, if antigen presenting cell is infected with virus, mostly dendritic cells happen, they express the viral antigen, which will bring the T cell even without any proper interaction in this case. So, both the cases, whether either it is a virus or bacterial antigen, both the cases T cell and MHC come together without or with proper interaction. So, if the proper interaction is there, so antigen recognition is there, it will come automatically, we do not need super antigen, but even if it is not there, they will come closer and interact and signal will go to the T cell for proliferation. So, this kind of super antigen, what they are doing, it is a general inflammation kind of thing. So, whole T cell population, wherever they will find interact, that T cell non-specifically will be get activated and proliferate. Okay. So, this is, and two examples are here. I mean, here one is staphylococcal enterotoxin, this one SE stands for staphylococcal enterotoxin, and other one is toxic shock syndrome toxin, and this is also Staphylococcus aureus, okay, also produced by the staphylococcal toxin. Okay. Both the cases, these two proteins can act as super antigen, many viral protein can act as super antigen, but how this evolution or this kind of interaction helps, or it is negatively interact, or they block the T cell. What is the effect of this production of super antigen by this bacteria or virus is not very clear in case of pathology. Okay, it is not known much, but this is a very common, and this super antigen can cause some general fever, okay, some, because it will cause lot of different immune reaction, and as a result we see some side expression like we can feel fever or something can happen. But scientists are scientists. As soon as they saw this, the super antigen is have a general capability or capacity to activate the immune system, in general the T cell will be activated, they started thinking how can we exploit this property and do something. That is where the knowledge of regular system, like even in biology, most of the wherever you say biotechnology kind of development or anything. So, biological system is doing something for their own survival, for their own fitness to the system, okay, because they have to fit themselves best so that they can survive so many genome. So, but scientists or you, all of you are welcome, just whenever you see something, any effect, if you start thinking how we can use this for our day to day life, maybe medical, maybe something else, okay. Not everything is, not all technologies are medical, not everybody is helping the production of medicine or developing drug or developing vaccines, okay. Something else also can be possible. So, there are many such examples, I am not going to go that. So, to do biotechnology, these kind of information, okay. So, this is the place of information, like there are super antigen. If I tell you what can be done, there are super antigen present in some bacteria, also some viral protein are also acts as super antigen, which induce the T cell population non-specifically. So, the overall T cell population. So, how can be used? So, I am giving, I mean I am telling you because it is already known. This is used during immunization. what happened? People used to give the whole bacteria. Okay. So, if you inject a proper antigen for vaccination, what will happen? That definitely it will go activate the immune system, produce some memory cells, which will remember next time if the disease, real disease happen, that will protect us. That is a normal primary and secondary infection. So, during primary infection, what we mix with the vaccine is that we will discuss when we will discuss the vaccine, some material which that particular antigen which I want to raise the immunity or the interested particularly, that antigen of interest is definitely there. Along with that, if I mix some super antigen, what will happen? If I mix some pure antigen that I would like to raise the immunity against, that definitely it will be there. Along with that, little bit this super antigen molecule I just mix and inject. What will happen? Super antigen will activate the immune system. Suppose somebody's immune system is going down or not good or not very effective to that antigen, that will not, we do not have to think about that, because this super antigen will activate the T cell, particularly the T helper cell. You can see both of them are reacting with MHC2. T helper cell will be activated. When all T helper cell will be activated, whole immune system will be very active, and that among this all possible T helper cells, some of them which is very specific to the antigen that I injected along with the super antigen, that will also be activated. So, more immune response against that antigen of interest will be much more, right. So, that is the reason why we use the super antigen. So, that is a kind of information in immune system exploited in the regular technology, like how we can, very simple information, but someone thought and it worked nice, and there are a lot of super antigen or super antigen type source we use during immunization or various immunization that is going on to induce the immune system as a whole, at least for the first time. Remember, we do not use super antigen every booster dose. Okay. Booster dose I told in the last. The first injection is a primary injection, and then second one injection is called first booster. So, first injection, then first booster, second booster, third booster, something like that. It is not the first one is the first booster. First one is the first administration of the antigen, second administration is the first booster. So, first administration time only we give this super antigen, but once it is already activated against that, next time onward we do not use it. So, that is the process of immunization, that in one of the class when we will discuss what is vaccine, how we can develop, that time we will say again this word, maybe little bit more detail. Okay. So, this is super antigen which is normally present or present in nature, but can be exploited for our own purpose or some development of vaccine or immunization process. Now, this is a kind of summary what we have discussed so far. Okay. There is no point of reading all this, again it is written here, but as just to mention here, if there is anything I missed also, the protein product of MHC class 1 and class 2 genes are highly polymorphic. I am sure you have no doubt about it right now. MHC polymorphism affects antigen recognition by T cell, influencing both peptide binding and the contact between T cell receptor and MHC molecule, which indicate that the polymorphism given only change a different color in the binding cleft. Okay, MHC 1 and MHC 2 I showed you the alpha chain only. Okay. So, that part. Alloreactive T cells recognizing non-self MHC are very abundant, that we just showed. Okay. So, and we also told that this one is responsible for rejection of the transplant organ or host versus graft rejection or reaction. Many T cells can respond super antigen, that we just discussed. Okay. MHC polymorphism extend the range of antigen and which other system can respond. So, polymorphism, importance of polymorphism. Okay. So, why this polymorphism evolved what is the mechanism, why not other genes, many things could have happened, but this polymorphism how it is helpful, that we already discussed, because different kind of and peptide we can MHC can fit into it. So, if you have multiple variety of peptide can be displayed. Polygenic, which is not written here. Polygyny is also important, because I have say 200 different genes. If one gene does not work, or by some infection or something happen, one or two genes somehow blocked, my whole immune system will not be 0. So, still I will have many other copies of MHC. So, multiple copy of MHC gene also very important, not written here, but it is we should remember. Okay. Now, the last thing is many proteins involved in antigen processing and presentation are encoded by genes within MHC. I am repeating again. Many protein genes involved in antigen processing and presentation are encoded by genes within MHC. This part we have not discussed yet. So far, whatever we are discussing about structure of MHC, how it looks, where is the difference between MHC 1 and MHC 2, hope all of everything you remember. So, when the question may come, right, in your exam, in different form, in different national level test, comparison is mad or matching, something, some way or some other way, the question can come, how they are similar or how they are different. So, compare means both you have to tell similarity and dissimilarity, and difference mean only dissimilarity, that I am sure you know. And when MHC 1 and MHC 2 will be asked, you have to tell both their structural similarity, dissimilarity, as well as functional similarity and dissimilarity, including binding to CD8 and CD4, that MHC 1 binds to CD8, MHC 2 binds to CD4, it presents the endogenous antigen or peptide, and it presents the external peptides or protein which is taken from outside. So, that we have already seen, but all these should be there. Okay. But this last line which is written in yellow, the many proteins involved in antigen processing, that we have not studied, maybe in the next lecture we will start that. Name is antigen processing. Okay. So, many proteins involved in antigen processing, how they are located in MHC, they are encoded within the MHC. So, let us see whatever gene we are told MHC 1, how they organize, this is the gene organization of MHC genes. You remember that the bar diagram of different genes, like their polymorphism is very high, some are very low. So, these are the genes actually DP, DM, A means alpha and B means beta. There are DQ, DR, okay, there are two different DR, beta gene, 2 copies and A, and these are the class 2. You see here it is class 2. So, this is one class 2 DQ, DR, okay. So, DP, DO is also there, one alpha is here and beta is here. And if you see the MHC class 1, this is there are 3 genes A, B and C, polymorphism is there. So, in chromosome it looks like this. This is for human, you see this is called HLA, I am repeating again, in human it is called human leukocyte antigen, and in mouse it is called MHC. In mouse also there are different class 2 genes, but why we are saying that different antigen processing genes are in between, because these genes LMP, TAP, which responsible for antigen processing are present within this region. Okay. So, which you do not know now, but we will discuss what is this TAP, what is this LMP, what is this TAP BP. Okay. So, if you go the detailed chromosome map of human, like human chromosome genome organization, if you see this, that was the cartoon and this is the same map. You can all these genes, all these are yellow here, like DQ, DQ, DR, DRB3, DRA. So, all this yellow part is MHC 2 genes, this is MHC class 2, which has this again tap 1, tap 2, clear. This tap 1, tap 2 is very important, some of you may know, we will discuss again. MSC class 3 I will come later. MSC class 1 genes B, C and A is here. So, A, B and C. There are some other genes G, F, they are important, but not MSC class 1. If you remember the other which we did not discuss much. MSC class 3 genes are actually very tricky. We do not have any MSC class 3 like MSC 1 and MSC 2, there is no MSC class 3, but there are many genes, like you can see the complement genes are here, which is you are going to learn very soon, that complement genes, okay, complement 4, and there are some cytokines like TNF, tumor necrosis factor, TNF, which is a cytokine. So, these genes are present in between this MSC class 2 and class 1. Okay, there are very important gene which is required for MSC activation or processing of antigen. So, these genes which helping this antigen processing or immune system and activated along with the MSC, they are known as, but they are not MSC directly like MSC 1 and MSC 2. They are together, all together, they are called MSC class 3. Okay. It is again I am repeating, MSC, it is MSC class 3 is not like a specific type of protein like MSC 1 and 2, there is no relation with the presentation or T cell interaction. It is many proteins which involved in immune system, cytokines, complement and other proteins which activate the different process of antigen processing. So, these genes are known as MSC class 3. Okay. So, in next class we will see how the antigen process and antigen processing starts. Thank you very much.